I want to dive deeper into the current challenges, and each one of you have mentioned a few throughout your responses to the first question, from operating costs to expensive consumables, logistics. I want to get more into that. So the current challenges around manufacturing, whether it be capacity, automation, lack of skilled expertise, training. Thomas, can you start us off here with some of the big challenges in manufacturing and the why behind them?
Yes, for sure. As previously mentioned, the current commercial cell therapy products are autologous therapies, but we all know that allogenic cell therapies are also being developed. And it's really important to make the distinction between both therapies because the challenges are not at all the same. In autologous therapies, there is no possibility to build stock. So one of the top challenges in autologous therapy is the time, because it's only when a patient has been identified to be treated that the manufacturing workflow can be initiated. That means that the patient must wait several weeks prior receiving his treatment, and during that time, his disease continues to evolve. So a gain of several days can make the difference for the patient. And one of the big challenges is that your organization has to fit with that time constraint. Another important challenge specific to autologous, as mentioned by Mark, it's really the manufacturing success rate. Because a failure usually means no treatment for the patient. In traditional drug products, you can control all raw materials and your batch-to-batch consistency is good. But in autologous cell therapies, the cells could significantly vary from one patient to another patient. And the manufacturing process must absorb must be capable to absorb that variability. And obviously, the manufacturing success rate is also impacted by the fact that usually the process is most of the time open, and the level of automation is very low. And so there is a high risk of human error or contamination. I could also speak about other challenges, like the chain of identity, or like the fact that autologous therapies are endemic in labor. But I would like to speak a little bit about the challenges of the allogenic cell therapies. So as I mentioned, so the big difference in allogenic cell therapies, you can build stocks because the cells come from an healthy donor, and you can start your production ahead of time, and the patient can receive the treatment as soon as he has been identified. And I would say the top challenge in allogenic, it's really the yield and scalability, because really the target is to produce billion and billion cells. And for the moment, the manufacturing technology is not yet ready to manage that quantity of cells. When we think about more particularly to the expansion step or the fill and finish, the cryopreservation and so on, it's a lot of the challenges in the allogenic therapies.
Thanks,
Thomas, I'll just jump in real quick and say I truly agree with two of the big points that you brought up. I sort of categorize the first one as the source of cells. Because there's additional costs that come with autologous, as Ludek was talking about within this, that, I think we often think auto single use or single lot, and then allo having the ability to serve, multiple patients. And then perhaps the next stage, which I don't know that we've really gotten to yet, is the use of even cell lines. And what's funny to me, or has been humorous to me at some level, is how we move initially in the field far more precise, trying to be more and more precise with what these cells are. And it seems kind of asking the question whether we need to be that precise. And some of it is actually driven by cost. Obviously, allo and use of cell lines have their own complication, have their own challenges. There are technical challenges going into the clinic and so forth. But the second piece that you spoke on, and I'd be interested to hear more about, is what you called scalability. And there's sort of two aspects to that, because there's the scalability if you're in an allo or a setting where you're treating multiple patients, and there's the scalability when it's single lot and trying to get your consumables down. And so for trying to get volume down, and most are still using Zuris and larger scale continuous flow systems. My question to you is how do you see the scalability side of this? The cell side of it has, I think there's a lot of technology going on, at least the cell source. But on the scalability side, do you see technologies that you find interesting in that space?
But yeah, as I mentioned, really the final target is to produce as much as possible with a single lot. Huh? So we are talking about hundreds and hundreds of billion cells. And for sure, we all know that there is we are not going to produce them with hundreds and hundreds of cell culture flasks, for sure. And so here really a big challenge is relating to the expansion step. We all know that some company, they start to develop and to work on some kind of bioreactor. And so, yes, it will be really key to have a technology that could affordable to manage that quantity of cells.
Yeah, I cannot speak particularly about, sorry, only brief comment. I cannot speak particularly about the allogenic, but about the autologous product and culturing of the autologous products. Of course, there are different solutions that you can use. Depends again on in which stage of clinical development you are and for what are the capacities that you need to fulfill. But in our case, usually, of course, for smaller scale studies, it was always better to do this more or less manually, but in parallel already to start the works on the automation and on scaling up and have a plan. Of course, this has to be coordinated with your clinical development because, of course, any huge investment at the start in phase 1 of clinical studies might be not reasonable because you can still end up with the product where the efficiency would not be met. So this has to be very carefully coordinated. But in our case, as I'm saying, it was always better for smaller scale projects to work more or less manually within our cell therapy facility, and then simultaneously within the same time to invest into smaller project targeting the automation and targeting some larger scale devices for future to be prepared for next stages of clinical development, right? Of course, yes. This is very difficult. It's always about robustness of your manufacturing process. In case your process is robust enough, you are probably already eligible to move it to some automated product. So as early as you start to work with some good and reliable partner targeting automation and scaling up projects, it's definitely better for you.